Hello and welcome to this presentation. I'm Caio Fonseca and I'm going to present the paper titled Predator-Prey Adaptive Control Algorithm for Exosome-Based Molecular Communications Glioblastoma Treatment. This is the outline of the presentation. I'm going to talk a little bit about the introduction, then the exosome-based molecular communication system model as defined by the Predator-Prey model. We are also going to talk about our proposed adaptive control algorithm and the simulations we performed in this paper, and finally the conclusions of this work. So in this work, we present an adaptive algorithm that has the objective of controlling the molecular communication system as defined by the exosomes diffusing through the extracellular space tissue by using the predator-prey model. The algorithm will be placed and implemented on an external brain-machine interface and will virtually model the treatment process. The reason we chose to use the predator-prey model is due to its equivalence to the exosomes being released from the organoids playing the role of predators, while the glioblastoma tumor cells play the role of prey. So the dynamics of the system is defined by the growth and regression of the tumor as a function of the exosomes transmitted through the molecular communication system. The molecular communication system in our application is established between the induced neurostem cells, which represent the transmitter, and the glioblastoma tumor cells, which represent the receiver. The exosomes being released from the induced neurostem cells are the information carriers propagating through the molecular communication channel, which in this case is the brain extracellular space. And in order to trigger the induced neurostem cells to release exosomes, the brain-machine interface will emit a radio frequency signal that will interact with these cells, making them release the exosomes. So this external signal will serve as a control input for the molecular communication system. In this paper, we chose to model these interactions as a predator-prey model. So the predator-prey model is a mathematical model that is really useful and efficient in describing the dynamic interactions in an ecosystem between populations, specifically populations of predators and prey such as foxes and rabbits. So from this perspective, we use the predator-prey model to describe the interactions for this molecular communication system where the exosomes plays the role of a predator, and their target, the GBM cells, represent the prey population. As a mathematical model, we can describe the predator-prey as follows. So we have two equations representing the rate of increase or decrease in their populations, and specifically we have the first equation representing the rate of growth or regression of the tumor, the glioblastoma tumor, and in the second equation, we have the rate of increase or decrease in the population of exosomes represented by the time derivative E. So each of these parameters, A, B, sigma, and D, determines the behavior of the model. So the more accurate these parameters are, the more efficient our model will be. A represents the growth rate of the glioblastoma tumor. B represents the aggressiveness rate of the predator population. Sigma is associated with the increase of exosomes due to the contributions from the interactions between the exosomes and the GBM cells, and the D parameter represents the rate of loss, or in this case, the half-life of the exosomes. So in this table, we present the parameters estimated for our model, and initially, we also normalize the values of the tumor size G and the population of exosomes E. So with these initial values, the G0 have a value of 1, and E0, the number of exosomes relative to the tumor cells, is 0.01. The growth rate of the tumor is estimated in 1.4% a day, and the aggressiveness rate is 1.4 per day, which basically means that 1.4 tumor cells are killed by 1 exosome per day, and the signaling factor sigma is estimated to be 0.7 per day, and the half-life of exosomes to be 0.7 per day. For our adaptive control algorithm, we take into consideration that the dynamics of the system is defined by the predator-prey model, and we use that in order to control the molecular communication system as defined by the exosomes and the GBM cells. So as we can see in the block diagram, the external signal will interact with the induced neurostem cells, releasing exosomes into the system or into the channel in order to get to target the glioblastomas or the receivers in this case. So that means that we can control the sigma parameter that represents the contribution factor due to the interactions between the GBM cells and the exosomes. And that also means that we can control the concentration of exosomes by controlling this input. For our control algorithm, we will consider two cases. The first case, we have an adaptive sigma that adapts according to the external signal, and the second case where we have a constant sigma. So in the first case, the algorithm aims to control the sigma parameter by changing the concentration of exosomes in the extracellular space through the amplitude modulation of the radio frequency signal. We can see in the equations below the radio frequency signal that will interact with the induced neurostem cells, the concentration of exosomes that will be as a function of the amplitude modulation of the signal, the proportional controller Kp, and consequently the updated value for the parameter sigma considering the proportional controller Kp. For our open loop control system in this case, the sigma is the parameter to control by the RF signal input, and the adaptive changes in the virtual tumor model will determine the adaptations of the sigma as a parameter. So with the new and updated value for sigma, we can also update the second equation in the predator-prey model that represents the rate of increase or decrease in the population of exosomes. So the equation 2 now becomes the equation 12, or simply, as we can see in the equation 13, very similar to the equation 2, with the difference being the updated sigma that adapts according to the RF signal. For the second case, the sigma parameter is now constant, so it is like a biological constant like the other parameters in the model. So the production of exosomes will depend on the amplitude modulation of the radio frequency signal in order to control the output of the signal of the system, which basically means the total size or the total number of tumor cells. So in order to mathematically model this system, we add the proportional controller, Kp, into the equation 2 as another term in the equation, as we can see in the equation 14. So the Kp will be a function of the external input, which means that increasing the amplitude of the signal will also increase the release of exosomes through the induced neurostem cells, which consequently increase the concentration of exosomes. So we can describe the proportional controller then as a ratio between the current concentration and the maximum concentration of exosomes times the maximum value for sigma as biologically defined. After having our models defined, we perform simulations for the molecular communication as defined by the induced neurostem cells transmitter, the propagation channel as defined by the brain extracellular space, as well as the receivers or the GBM tumor cells, as we can see in the diagram. The exosomes released in their diffusion in the extracellular space can be modeled after the solution to Fick's second law of diffusion. And based on that, we reconstructed a portion of the brain extracellular space as the spaces or the gaps between the cubic cells, as demonstrated by previous works by Tao and Nicholson. And in order to perform the simulations on the exosomes diffusion through this medium that represents the molecular communication channel in this case. So the equation determines the concentration as a function of time in radial distance, as we can see here in equation 16. The N0 represents the initial number of molecules being released. D is the diffusion coefficient, or in this case, the effective diffusion coefficient, and R represents the radial distance. But having the interaction of the external signal with the induced neurostem cells results in more or less exosome release into the extracellular space, which in turn increases or decreases the exosome's concentration. So taking this into consideration, we model the concentration as a function of the amplitude as well as a function of the distance and the time. And we can see this in equation 17. So now, based on the equation 17, we can simulate the concentration of exosomes. So we perform the simulations for a distance of 5 microns and a diffusion coefficient of 15 times 10 to the minus 6. And as we can see in the graph, we have the concentration as a function of the amplitude signal, And the more we increase the amplitude of the radio frequency signal, the higher the concentration of exosomes will be in the extracellular space, as we can see illustrated in this graph. We also performed simulations to observe the molecular diffusion through the reconstructed brain extracellular space made of cubic cells. So this reconstruction is made by putting together cubic cells in a volume where the gaps or the spaces between the cubes represent the brain extracellular space. The volume of the extracellular space in this reconstructed volume is about 20% of the total volume, which is in agreement with the volume fraction of the extracellular space in the human brain. Then, if we integrate the concentration equation over a cubic volume, we can obtain the equation that represents the number of molecules that will diffuse over time inside a cubic volume of length size 2a, as we can see in the equation 18. So using the reconstructed brain extracellular space, we simulated the diffusion of molecules through the tissue. The simulations had 1,000 molecules being released from a point source and a diffusion constant of 10 to the minus 6. The reconstructed neurotissue was defined by 1,000 cubic cells with a length of 0.5 microns and a spacing between them of 42.9 nanometers, which makes 20% of the total volume. The number of molecules were recorded using extra cubes that did not interact with the molecules, so they served only as sensors or sampling box to record the number of molecules in that specific volume with length sizes of 2, 3, 4, and 5 microns, as we can see in the graph. And each curve represents the number of molecules inside the sampling box diffusing over time. We also performed simulations to demonstrate the behavior of the model as defined by the predator-prey system. The simulations illustrate the tumor growth and decay through time, as we can observe in the graphs below. So the first graph represents the tumor growth as a function of the sigma parameter. And for the value of 0.7, we can see the steady state scenario where the tumor does not grow nor shrink. And for other values of sigma, we can start to see the oscillatory behavior of the tumor growth as defined by the predator-prey model. Another important thing to notice is that it is not only a matter of increasing the value of sigma in order to be more efficient in eradicating the tumor. So for the value of 1, the tumor gets close to being eradicated in a little bit more of 10 than 100 days, but it starts to grow again, reaching almost 3 times its original size before being eradicated after more than 400 days. So it is fundamental that the tumor is completely eliminated, otherwise it can start growing again. The best value simulation was for sigma equals to 1.1, where the tumor was eradicated in less than 200 days. So this analysis is fundamental in order to find the values of sigma where the elimination of the tumor will be more efficient, requiring less time to eradicate it. The second graph is the simulation showing the tumor growth with the controller applied and the sigma adapting to the system. So we can see that for an amplitude ratio of 0.3 and 0.5, the tumor growth oscillates and does not get eradicated in less than 200 days. For 0.5, it gets close to being eradicated, but it starts to grow again. For values of 0.75 and 0.9, it gets eradicated very fast in less than 25 days, so the controller works very efficiently for these values. And in the third graph, we can see the tumor growth with a fixed sigma value of 1.25, which was the worst value in the first simulation, and the controller applied to the system. In this case, the controller is also very efficient in eradicating the tumor for all values of amplitude ratios. The time for eradicating the tumor can also be different in the case for different values of sigma, since the proportional controller does not change it. However, it will still be efficient in eliminating the tumor, as we've seen that this case, we used the sigma value that was the worst case in the first simulation. So in this paper, we modeled the interactions between the exosomes and the GBM tumor cells as a predator-prey system, where the exosomes play the role of the predator population and the GBM tumor cells represent the prey population. We present our adaptive control algorithm for brain-machine interfaces in the context of molecular communication system theranostics and targeted drug delivery, presenting also our analysis for 2 possible scenarios for this control system. We also consider the molecular communication system established between the exosomes and the GBM tumor cells, as well as the propagation of the exosomes through this medium, through the extracellular space channel, and how this influences the system. The results from the simulations were able to show that the controller is efficient in eradicating the tumor by drastically reducing the time. And also this work paves the way and create opportunities for novel biotechnology solutions to tumor theranostics using the principles of molecular communication. And for our future work, we have the objective of extending our adaptive control algorithm to a closed-loop control system by using the response feedback from the messaging neural stem cells so that we can adjust the input signal and consequently fine-tune the controller in order to have a real-time control signal. Thank you. If you have any questions, please feel free to contact me or any author of this paper.